will be emailed to participants approximately 30 days following the webinar. Today, Dr. Rumila will address next-gen sequencing and intro to clinical applications. Allow me to introduce Dr. Andy Rumila. Dr. Rumila went to medical school at the University of Illinois in Chicago. She did a pathology residency as well as a fellowship in molecular genetic pathology at Mayo Clinic in Rochester, Minnesota. She is particularly interested in hereditary cancer syndromes and targeted therapies for cancer. Dr. Rumila?
Greetings. So today would like to discuss a brief presentation about next-generation sequencing and an introduction to some of the clinical applications of this technology. I have no relevant personal financial disclosures or off-label usage. The general objectives for this talk are to gain an understanding of what next-generation sequencing is, what makes it different, and what makes it the same. Explain the range of potential testing possibilities, including the differential between germline and somatic testing, and then to give examples of the clinical utility and the challenges of genomic testing. So let's start off with a simple question. What is NGS? Well, technically, next-generation sequencing. But what exactly that stands for, what that means, is basically it's a category of technology that includes a number of different platforms in a laboratory that create or generate large numbers of nucleic acid sequences. So basically everything that's not Sanger sequencing or the older styles of pyrosequencing, but just the large high throughput or deep sequencing boxes that are now available from a number of different companies. This is a style or a type of sequencing that is also heavily dependent on bioinformatics. So there's a lot of information regarding software programs and tools designed to synthesize the data, the actual data coming off of the sequencers in a number of different ways. We will touch on this part lightly during the talk, but that's literally hours and hours and whole courses in and of itself. Okay, so what makes NGS different from Sanger? Well, basically the nuts and bolts of it, it's the sheer volume of the data. It's some back-of-the-napkin calculations. Per run, if you take your average, say, ABI 3700 96 head capillary electrophoresis, plugging out, your best sequencing run ever, a good 600 bases, your average run, if all 96 wells work perfectly, you're probably on the order of about 60,000 bases. Whereas depending on the instrument, and yes, don't hold me to the exact numbers, there's lots of variation between the different boxes. But when you're talking next-generation sequencing, you're in a whole different ballpark, where you're at about, say, 6 billion reads per run, depending on the instrument, and each run being kind of on average maybe 200 bases or so, you're at over 1 trillion bases per run. And runs on the different instruments, say it's a 24-hour run, that's an awful lot of data that you're generating. So the sheer volume is pretty much the fundamental thing that makes NGS different. And for that matter, different and, a disruptive technology in that it changes the way we look and think about a lot of diagnostics and a lot of laboratory medicine. So the approach to sequencing also makes it a little different. In the past, on the left-hand side of the screen, you kind of have the targeted sequencing, what we generally think of as Sanger sequencing. You have your DNA strands, and the little arrows representing primers were designed to a specific region of your DNA. You knew that in advance. There are ways to get around that if you didn't know the exact sequence, different ways of cloning and whatnot, but by and large, you knew what you were sequencing, and then you generated sequence from that, as pictured below. Well, starting with the sequencing of the human genome on the right side, we kind of had to change that approach when you were talking about sequencing the entire human genome. And so began kind of that shotgun or larger scale sequencing approaches. And so as you go through the different images, we're not using back libraries anymore, but the general schematic still applies, where you're taking your germline DNA or your whatever DNA source you're testing, shredding it up into pieces, little clones, and then just sequencing them. And then as you can see from at the bottom where it says shotgun sequence and assembly, you're taking those shorter sequences and bioinformatically putting them back together. Okay, next thing that makes NGS different. Well, it's a lot cheaper and a lot faster, and you generate a lot more data. So going back to the whole idea of scale, when you look at Moore's law and kind of how that would trend in terms of the coming down of cost over time for sequencing, we kind of put Moore's law to shame because the community has really dropped off that graph. And you can see starting at about 2008, those numbers really started to plummet. And this graph only goes to 2011, so you know that per base, this is getting incredibly cheap to generate the sequence. Now let's talk about the process on the next slide. And you can see it's not just the sequencing. And when people often say the cost of sequencing, that last graph that we just had, they're talking about the purple box, the generation of the sequence. And that's not the whole story, though. When you're talking about clinical application and making useful technology and a useful test out of these skills, you need to see the entire picture, from designing the clinical test to how you manage and interpret the data. And so that really changes the overall scheme of how this type of testing works. Generating the sequencing itself, as you can see over time, will become a smaller and smaller fraction of the overall cost and of the overall process that goes into a clinical test that involves this technology. And speaking of the workflow, let's get into some details about that workflow. Sample collection and processing. You need to either draw the blood or obtain the tissue, whatever your target matter is that you're going to sequence the DNA from or RNA from. And then there's actual generation of sequencing. So this is, at this point, is where you've extracted the DNA from your source, you've built your libraries, you've put it on the machine, and it's generated the bioinformatic files, usually BAM files, FASTA files, that then get fed into some of the bioinformatics, which you start that right away. Sorry, I couldn't get my little notes on the right side to come up. The primary analysis of it, you align all those short reads that are generated by next-generation sequencing into one cohesive sequence. And then you need to compare that to something and identify what the variants are. And this is where terminology gets a little tricky. At this point, they're all just variants. There are differences that are identified between the sequence that you've generated and whatever reference you're using. And then comes the next step. So for variant interpretation, you need to take those variants and then identify, well, are they polymorphisms? Are they alleles that are pathogenic? Are they newly discovered rare alterations that we know very little about? That variant interpretation, really the annotation of the findings, are really what becomes crucial when you're talking about a clinical test. What's the significance of that alteration? What can you do with that alteration? What is it telling you? And then depending on what the test is, what the point, what the clinical indication was for the test, how does that affect your patient? And then, of course, even once you have all that in the lab and you know that, you still have to then create a report and communicate that to the individuals who are taking care of the patients. And as I already mentioned, each platform has its own chemistry for generating sequencing. And so basically, the next few slides will be a very brief overview, not designed to be for or against any one of these different companies. There are pros and cons to everything, of course, but like I said, I'm not trying to promote any one, just trying to show that there are lots of different technologies and they take somewhat different approaches to next generation sequencing. So I think kind of comparing them and looking a little bit of some of those processes, the technologies themselves, is a little bit helpful to give you a better idea of this basket of technology that people talk about as NGS. So for Illumina, you have basically it's sequencing by synthesis. So you're generating the strand as you sequence it, much like Sanger does. And you have 4 different color bases. As you add them on, you have the little photo capture that analyzes what bases are being added on at each particular place on a cluster which is rooted on a solid surface. And then you can have many, many of these clusters, millions of these clusters on a slide, and you can then just bioinformatically tally up that sequence, put all those fragments together, and generate your sequence. For helicose, this is slightly different. Same idea as far as sequencing by synthesis and having your clusters generated on a solid surface. But here you've got a slightly different technology where you're looking at different bases with the same, essentially, color or fluorescence that are sequenced through one by one, but again, you can read them, and you can read them off, again, and you can see the picture at the bottom left. Again, with the photo, with the cameras, you can read the little spots and count off what those bases should be. Again, bioinformatically put that all together. For LifeTech, this is a slightly different approach. It's 4 different colors, but it's sequencing by ligation. It's one of the bead-based platforms, and there's a different construct that's used that identifies 2 bases at a time. And it's not necessarily sequential bases. You can tell from the graphics that there are little gaps based on the ligation cycle and where your primer is set. So the primers are designed to be offset by a base. And so every time you go through a cycle, you start counting off by that base. Bioinformatically, you can put them all back together, and you can see at the bottom, each base actually gets represented twice by the little dimers. And you can put those reads back together and, again, generate your entire sequence. Roche, you can move on to, again, single base, but again, it's single base by pyrosequencing, except on a much larger scale. So you've got all your little nano wells, and again, an emulsion-based, bead-based PCR, where each little emulsion has all its ingredients, and you're generating a fluorescence every time a specific base is added. And you can tell at the bottom, then you can just read off that sequence and generate your reads. For PacBio, it's a slightly different approach. Here you've got your enzyme attached to the solid phase, but once again, you have a 4-color sequencing by synthesis. So as you're sequencing that strand, which is in a fixed place, a single strand in the well, you're emitting the light, and then that's what's generated compared to baseline. So again, you're getting that imaged, and each piece that's getting imaged, you can see as each base is added and generate a sequence at each particular location. Nanopore is a little different. It's enzyme dependent. Some of the initial platforms had an exonuclease where it's sequencing by deconstruction. That's not, the current platforms that are available are using a DNA polymerase. So again, you're just generating the sequence and you're putting the strand as it synthesizes through the pore. Again, the idea is that you can identify the base as it goes through that pore by how it affects the charge through the pore. And staying on the charge idea, Ion Torrent has a different approach, whereas it generates its sequence, again, in its small single amplicon pore, you can, it's sequencing by synthesis here as well, and you can also, as it releases, as each reaction, as each base is added on, it releases a hydrogen atom, which is then detected. And so you can tell by which base has been put through the system whether a base was added or not. Okay. So that's a really, really high, quick breeze through some of the common NGS platforms. And then comes analysis and interpretation. Again, so now you have numerous reads. Bioinformatically, you can compare the 2 pictures. On the right side, you have your average on your left side, you have your average chromatogram of Sanger sequencing, and you can see an alteration there in the middle. On the left-hand side, or on the right-hand side, you can see there's a lot of alterations, and each one of those green and blue bands or cigars represents another read coming from an NGS sequencer. And you can tell from that there's a lot more reads in there, and you can tell a lot of them are identifying that same alteration. So it's a different way to visualize it. And then you still have to go back and identify, well, what does that alteration mean? What test is being answered, what question is being answered, and how that applies to the patient. Sorry about that. So as you're looking at that data, something else pops into mind that I want to discuss, and that is the impact of read depth. Because you're generating such data, and you can read the same sequence or the same particular base so many times as you're reading, as the different reads go through and are compiled by the bioinformatics pipeline, you get a very different sense of your detection rate. It changes quite a bit when you're comparing it to the classic Sanger. So on the left-hand side, you see kind of what happens as the percentage in say, a tumor, where you're not necessarily expecting a 50-50 heterozygous alteration. You see the far most left one. You can see there's a pretty decent change there from G to A, and it's probably close to 50-50. But as you move to the right, it gets a little harder to call that. The little alterations are not quite so prominent. Perhaps the tumor percent is less than 50, or perhaps the the tumor cells don't all carry the same alteration. And so it gets to a point that it's very difficult either for the software or for the human eye to call that. And when it comes to Sanger sequencing, that probably starts happening at about 20, 25%. So there's a lot of heterogeneity that you might not be seeing, particularly in tumors. And so it's really not the ideal technology, which is where next generation comes in. This is, I mean, there are many other technologies that are also very good at and have very high sensitivity and can detect a very low copy number, and NGS is one of them. You can see from the two pictures on the right, there are a couple different examples that show alterations. The middle picture, you're talking about a 60-40 ratio, which is, you'd see that pretty comfortably on Sanger. You can see it pretty comfortably on NGS as well. And the thing to note is, right after the C colon, I've got 16,000 times that that particular base has been read. My total count of reads is close to 26,000 on this particular test. So that's pretty deep reading. I can see lots and lots of times that it's a pretty real alteration. You can also see that there is some variation with all those reads. There are some chances for mistakes. You can see that there's some insertions and deletions that are being called that are not real. There's a couple G's that are being flagged in there that, again, are not real. It's less than 1% of the overall read count. And then you can compare that to the one on the right, where I've got one alteration, or one base, being 87% of the reads at that one particular base, and yet it still sees quite comfortably the 13% of the reads that have an A. Again, I've got over 7,000 reads for this one particular base. So it changes how comfortable you are and what your limits of detection can be. And this is where knowing the platforms and knowing the error rates of the different platforms becomes important as you're setting your limit of detection for clinical assays. And this feeds us right into, well, what makes this like any other test? I mean, we're trying to develop and perform assays for clinical laboratories. So what are we using this technology for? Well, there's screening tests. Newborn screening is definitely establishing itself using this technology in a whole different way. Sorry, not newborn screening, prenatal screening, looking at chromosomal abnormalities by now having the capacity to count sequences in mom's blood and count them so efficiently that we can actually count the fetal sequences in mom's blood. So that's something that the deep sequencing and the many, many fold times we can count each base is allowing us to do and expand the testing in the screening territory. For diagnosis, there's any number of examples that would apply for this. Looking for hereditary syndromes, looking for abnormalities that maybe would have been very difficult, or there were many possible genes where the alteration could lie, which was not necessarily feasible or very easy to do on Sanger, which becomes much more feasible by next generation sequencing. Prognosis. And this is where we start to cross over a little bit into the examples I'll have on the somatic or tumor side. Alterations that we know either portend a good or a bad prognosis for various tumor types, which ties in also very well to the therapeutics. But in the therapeutics, I would say it's not just colon cancer, or not just cancers and somatic alterations that have an impact. When you think about all the pharmacogenomic examples and implications to clinical health and management, if that's all helping people determine what's the right therapeutic and what drug to give the individual. So in my mind, those all fit under the therapeutic umbrella as well. And monitoring. We've known for a long time now, and it's been well established in some of the hematologic disorders, where you monitor for relapse, and you monitor it different ways, whether it's histologically in bone marrow, or looking at fish, or going to the molecular markers, where how low can you detect when something is starting to come back, when a clone is starting to grow back. So that's also important. And these are all applications that we've used non-NGS methods for in the past. So really, we're still answering and asking the same clinical questions, which is why, depending on how you look at this, it's just another chemistry for obtaining a series of nucleotides. The methods for bringing tests up in clinical laboratories are the same. You want to go through your developmental experiments, optimize the technology, optimize the test, making sure your clinical utility, whether it's reproducible, your reporting range, all the different elements that are requested and required for other tests apply to this just as well. Subject to regulation, there's a lot of discussion about regulation, but in the end, it's a clinical test that you're applying to patients. It's something that falls under, CAP, CLIA, FDA. FDA is looking at, NGS platforms to approve as well. So this is all something that fits into the same framework and model of the clinical lab. All right, so we've already started talking about the various examples for clinical testing. And personally, I like to break things down, starting off with somatic and germline. For the somatic, generally you're getting heterogeneity. You're getting 50-50. So you're heterozygous or not. We know that there's some mosaicism, so we know that that's always something to consider on the hereditary side. And on the somatic side, even though you're testing tumor, in my mind, you start to get a little bit more complicated because we'll talk in a little bit about what kind of tumors you have. Is it fixed? Is it fresh? Different fixatives have different effects on DNA. And when you get to the larger panel, sometimes it's useful to have the germline as well in order to cancel out or filter out the polymorphisms that aren't relevant to the tumor but that are different from, from one individual to the next. I'll go through a few examples. And again, just to kind of show the progression as far as genomic profiling on the somatic side, we're looking for the same types of things we've known about for a long time. And we've gone from PCR-based assays and FISH probes, single gene tests to panels to having the capacity to do exome and genome-wide analysis. But again, when we get into the interpretation side, this is where you start to see variants and things that we really don't know what to do with even in the panel stage. So let's look at a couple examples. First patient, an individual with metastatic angiosarcoma, has had multiple drug regimens, is not responding well. They're looking for what else can we give this individual to try and control their cancer. Well, so a next generation sequencing platform was ordered on paraffin-embedded tumor tissue. This is very standard. We do single gene assays. There's numerous platforms and panels that are available out there. And a particular fusion was identified in this case. Okay. It was classified as activating, so as people went through and looked in the literature, they found that rearrangements of ROS1 in non-small cell lung cancer has been FDA approved as a predictive of response to a class of drugs, including crizotinib. But wait a minute, it's been well studied and is approved in lung cancer, but this isn't lung cancer. Turns out, rearrangements for ROS1 in particular have been reported in glioblastomas, a number of other carcinomas, but not really a whole lot in the sarcomas. Okay, so what's next? Does this apply just as much to the sarcomas? Are we okay to give the same drug? What can we do? Is this going to answer the clinical question that was being asked? Can this individual, what other drugs can we give this individual to try and fight their cancer? So it turns out for this particular gene, what's being looked at in terms of predictive of drug response has been the FISH assay. And here we have two examples. It's a break apart FISH strategy. So on the left-hand side, you can see the red and the green are right next to each other. They're together, as they would be in a normal chromosome. And then on the picture on the right, you can see there's one fused one, the green and the red together. And then you can see in most of the cells in that field, you also have a green and a red that are in very different parts of the nucleus, suggesting that there's been a break apart, a rearrangement of the ROS1. Now in this particular field, you really can't see what it's rearranged with. You can't see what its partner is, but it shows that there's a rearrangement. And this is typically the test that's currently being used to identify patients whose tumors would be eligible for this particular targeted drug. Now, this is very commonly ordered for lung carcinomas, but not really for angiosarcomas. So in this particular case, the biggest difference that the panel made was that it highlighted something that would not necessarily have been thought of right away for the particular tumor type that this individual is faced with. So in a sense, by casting a wider net, we were able to identify something that might not have necessarily been ordered for this patient otherwise. Now, the next question is, okay, well, that's very fine and good, and I can confirm that ROS1 is rearranged using FISH probes. However, is that really being expressed? And so in this particular case, we have some RNA sequencing data, again, by next-generation sequencing, that can speak to the idea of, or speak to the question, is this being expressed? Is it functional? And trying to address that question of, is it functional? Is it something that we could potentially use to help this patient? And you can see in this particular schematic, the part of the gene that is involved in the fusion and is thought to be expressed is expressed at a much higher level, represented by the kind of orangey pink line, than the part of the ROS1 that's not involved in the fusion or is not thought to be transcribed as part of the fusion. And so, again, you're trying to answer the question of, is this relevant to my patient and can it be used for treatment? And so the idea there is that this is something that the patient might be eligible for, and it turns out that there are actual trials out there that he may be able to qualify for given this evidence. Next example. So patient has metastatic ovarian carcinoma, again, has had multiple chemotherapeutic regimens, and again, they're trying to figure out, what do we give our patient next? A next-generation sequencing panel was performed, again, on paraffin-embedded tissue. And the most interesting finding in this case was an ALK alteration. So it's not a rearrangement. It's a missense mutation that leads to an amino acid change at a particular codon. Okay, well, if you look at in silico data, it's listed as potentially damaging, but it actually hasn't been reported a whole lot. There's not much in the literature. You look at population databases, there's not much known. It's not a common alteration that's found in the population. It's not just a SNP. So what do we do with that? Now the question, again, arises because there's an FDA-approved therapy available that targets ALK. However, this isn't the alteration that's generally used as a predictive of response to ALK therapies. And again, not so much in ovarian cancer. This has been really more thoroughly studied in lung cancer. So you're trying to ask the same question. What other drug can I give my patient? And you're faced with the same follow-up question, even though now you have a molecular answer. Well, what's next? Is this alteration going to be enough to allow my patient to respond to this drug? Well, the typical tests that are currently being performed, what's FDA approved to be performed for predicting anti-ALK therapy response, is FISH, again, a break-apart strategy, and more recently, an immunohistochemistry result. So we went ahead and stained them and ran the test, and you can see that there really wasn't a whole lot of good fusion or good rearrangement identified by FISH. It was below the level that would be considered for calling an abnormality in the ALK locus by FISH. Well, okay, so the FISH really didn't confirm that, but we really weren't expecting a rearrangement because what we had seen by sequencing was a missense mutation, not a rearrangement. But then we did immunohistochemistry. Okay, so we're seeing some expression. It's somewhat patchy. There's some staining. It seems to be somewhat apical, but not really entirely fitting the pattern we would typically expect. But again, is this something that's demonstrating something that's caused by this particular missense mutation or not? And this is where really we don't know. And that's something to think about as the panels expand and we're trying to interpret data. We don't always have the answer to the clinical question. Even though in this particular case we found an alteration, it really wasn't anything that we thought was enough evidence to say that, yes, this would be an active tyrosine kinase or an active enzyme or an active molecule that could be targeted by the available drugs. And that's something to think about when you scale up to whole exome and whole genome. The more you look at, the more variants you can find. Again, for somatic testing, you can filter out some of the germline, but remember you're trying to answer a clinical question when you're in the labs. What's actionable? What's predictive or prognostic? Or what gives you an answer? And sometimes we have found that it can be helpful in getting into trials. Sometimes it can find unexpected for a tumor type, like the previous example. And sometimes it becomes a bit of a challenge in terms of interpreting it. And you have to turn to literature and other data to try and inform some of those findings. But again, it's not a clinical test at that point. So it becomes a very difficult interpretation. RNA is also coming into the clinical mainstream. So we've been doing RT-PCR for a long time. But again, as you scale that up to next generation sequencing, you're really looking at not just any one particular translocation, any one particular fusion that you know about that you're looking for. Now you're just basically asking the question, can I find translocations? Can I find evidence of alternative splicing or altered gene expression? And they all come with interesting interpretation and bioinformatic questions as you try to analyze the data. And again, for somatic tumors and somatic indications, you also start facing the question of fresh frozen, what fixatives, RNA later, is it in formalin? You know, different fixatives have different effects on nucleic acid. And so how likely is your target nucleic acid going to be to survive the processing? And we'll talk a little bit more about that in a few minutes as well. So bioinformatics, different pipelines, different analytic tools. How do you align the data? How do you look for certain events and then interpret it? Comparison to gold standard also becomes a bit of a challenge because now, especially as you're looking at expression, you've kind of changed the playing field and the part of the dogma that you're looking at. It's not just the fusion. It's not just looking at the rearrangement of the DNA, but is it in frame? Is it something that can express a functional protein? And so in some cases, comparison to gold standard can be very difficult, or even just how to compare to normal expression can be a challenge. Okay, so let's talk about that quality of the target that you're trying to read or sequence. And so between RNA and DNA, a very different playing field. DNA is very stable and a very hardy molecule compared to RNA in many ways. And this is very true, again, when you have paraffin embedded or even fresh tissue. The quality of the RNA in that material starts to degrade much, much faster than DNA. And so you start looking at other tools or other assessment mechanisms to see how good your template is. And one score that's commonly used is the RNA integrity number. And here we're looking at a little graphic that shows as that number decreases from 10 to 0, the coverage that you have in this particular gene starts to drop off. So now keep in mind this is an example using a poly-A tail capture. So basically we're trying to pull out the RNA based on it being a messenger RNA and having a poly-A tail. And so your 3' will have the best representation because that's what's closest to the tail. And then as you move 5' to the left on your screen, you can see that there's progressively less and less reads coming from those areas of the gene. And as the RIN score drops off, again, you can see that that gray space, this space up here representing the pileup of reads, decreases significantly. And the more 3' in the gene you get, these little representations of the reads really drop off. When you're looking for fusion, you're looking for, of all those reads and from all those different regions, you need to look at what are your supporting reads. Reads that span the breakpoint, or reads where one half comes from one place and the other half comes from the other part of the fusion. And so as you dilute that tumor percent and your tumor has more and more normal cells mixed in with it, or you have fewer and fewer reads because of degradation of your RNA, you need to be very cognizant of how many supporting reads you can still find. And the way you calculate limit of detection pulls in a few more variables. And so let's take a very common example using BCR-ABL. So pretty well-known, well-characterized fusion. And something that's very basic and very fundamental to the identification of particular neoplasms in the hematologic world. Well, it turns out this is a pretty big gene. There's a lot of kilobases here. And if you're trying to get intact RNA, how many reads you have as you move more and more 5' can dramatically decrease, even in a good quality sample. And so of the reads that you have in the particular region that you're targeting, you have to have enough representation that you're going to be able to detect and have confidence that the alteration you're detecting is real. And you can see in this particular case where you have a RIN of 7.1, the 5' end of the able part of the fusion has a lot of reads, has 85 reads. But as you move more and more 5' at exon 2, where your breakpoint, where you're expecting to find your breakpoint with BCR, you're only down to 19 reads. And so if all the cells you have in your sample are neoplastic or part of that CML, then you would expect that the majority of the sequence you have is from those abnormal cells. You'd expect a good percentage of these reads to be abnormal. But if your sample only has 20% of 19 reads, would be very difficult to be confident in detecting that. So let's go on to another example. Let's look at expression. How do you normalize expression data? It's more than this or less than that. Metastatic lung cancer, well, you take normal lung and compare. Is it more or less expressed than normal lung parenchyma? Okay, so for that solid tumor, maybe it's not so complicated. But what about breast cancer? What's your normal comparison for breast cancer? Breast epithelium? Well, in your normal breast tissue, that's actually a small percentage of the cells that are in normal breast tissue is the epithelium. So it's maybe not always so easy to get a normal sample to compare to. And depending on what you're looking at, there can be very significant differences in what's normal expression for a tissue from tissue to tissue. So comparing between other tissues or other normals may or may not be significant for what you're looking at. Let's take our example, seeing as how I've already mentioned breast cancer, ERBB2. So looking at reads per kilobase per million, you can see that in this particular sample, you've got a lot. Your red bar on the graphic on the left on the waterfall plot shows you've got a lot of reads, and far more than all the other samples you're comparing to. But what are all these other samples? You can display it any number of ways, however is visually appealing to you, but in the end, you have to know what you're comparing to. Because in the end, you want to know, how does this compare to IHC? How does this compare to FISH? Is this change in expression enough to say my patient will or will not respond to the anti-HER2 drugs that are available? Because in the end, that's what you're trying to answer for your patient, right? So there are other technologies, other standards out there. How does this compare? How does it compare to breast? What if all these samples down here, what if they're breast cancers? And this looks like there's a big increase in expression. So we know that this would be positive, or how this would correlate by FISH or by immunohistochemistry, which are the two methods we're currently using for determining response to HER2, to anti-HER2 drugs. What if all of these are normal, either normal breast epithelium or normal any cell in the human body? Is that a significant change? So comparisons and how you calculate these are important things to consider when you look at this data. It's not just sufficient to generate the data. Would you give the drug? I mean, at what point and what comparisons do you need to have in order to generate a clinical actionable response? Would it be different for different tumors? Okay, next one, example 4. Looking at a different one, so one that we know a little less about, FGFR3. Again, you see there's lots of comparator samples here. And again, the one sample you're looking at has a much, much higher expression. So again, is this number significant? Does this mean something in terms of predicting response? It turns out in this particular example, an array had been done. So we actually are able to compare that data to DNA gains and losses. And it turns out that this is a region that's gained, that shows some gain in chromosome 4. Okay. Again, is this something that is predictive to respond to the drug for this particular tumor type? And so those are all questions that need to be answered. It's not just generating the data, it's knowing what to do with it and how it applies to the question at hand. So in this particular case, it mattered for the patient what tumor type it was, what data is out there. Are there clinical trials that are currently looking at this? Okay, so I've given you a couple examples, and now let's talk a little bit about tissue. what is needed and why. There's lots of ways to get tissue, and tissue comes in all sorts of fixatives, and it can be processed in a number of different ways. In the end, what you're looking for is sufficient quantity, quality, and when I mean quality, I mean concentration for the assay and the length of the fragments that are appropriate for the platform you're using to provide a good, reproducible, reliable result. Question between fresh frozen and formalin fixed paraffin embedded tissue is one of the natural questions that comes to mind. Well, it depends on your platform. If your platform is something that's giving you very short reads, well, paraffin embedded may not be so bad, because the process of formalin fixation and paraffin embedding tissue basically results in usable DNA length of about 200 bases. If you're lucky, maybe getting a little closer to 300 bases, but really not much beyond that. If your platform really requires a longer length of intact DNA or RNA, you may need something like fresh frozen, where you haven't exposed your RNA and your DNA to the disruption or the cross-linking or whatever fixative damage has been caused in your other fixatives or paraffin-embedded material. Tumor percent is something to think about as well. I'll show some pictures about this. So let's think about the general processing of tissue. Tissue is obtained in some way, either a surgical resection like this, or a biopsy, a swab. Somehow or another, neoplastic cells are obtained from the body. And then they are, in most cases, kind of the traditional approach is they are paraffin embedded. They can be fixed by a number of different fixatives. Formalin, 10% neutral buffered formalin is probably the most common. It is not the most damaging, and it's not the best either. It's sort of in the middle of the pack. There are far worse fixatives, and there are far gentler fixatives. But the vast majority of tissue ends up in a block or a slide. Blocks eventually get cut, and tissue slices are put onto slides so we can look at them. Those are the pictures up above. But as you can very quickly see, these tissues are not created equal. You can see there's, on the top left, there's a large area of tumor, and there's a large area of normal tissue. The tissue on the left is very small, small fragments of tissue, but it has a good tumor percent written on that slide. Okay, so right off the bat, you get a sense of the quantity of DNA you may have available to work with. This tissue, you've got lots and lots of nuclear material. Here, not so much, although the percentages are both fairly good. Now, as you look at the bottom fields, they kind of change the story around a little bit. These are not the same, these are not from the slides that are above. These are other examples. But you can see here, these larger cells, these little nuclei here, these are the neoplastic nuclei. The smaller purple dots and some of these pink fibrotic bands with the rare stromal cells in there, those are the benign cells. So of the nuclei that are present in this sample, the vast majority of them are neoplastic. This example, on the other hand, even though you kind of get the sense that this may be a larger fragment of tissue, you have a lot of these really dark purple spots, the lymphocytes. There's tons of these. And your neoplastic cells are lining these spaces. So you have far fewer neoplastic nuclei than you have benign nuclei in this particular sample. So even though it's a very large sample, the percentage of cells that you're actually looking to find the abnormalities in only represent a small amount of the DNA that's present in the sample. So again, now you go back to what technology you're trying to use this tissue for. If you're using this tissue for, say, fish, where you keep the architecture intact and you can look at what cells you want to interrogate with your fish probes, you can identify them a little better, it may not be much of an issue. However, if you're looking for a platform, if you're using a platform that requires extraction of DNA or RNA, and you're only able to identify, you're down to a sensitivity of maybe 1 or 2%, maybe even that's not a problem, because what percentage of all the nuclei that are here are malignant? Now, what you have to also keep in mind for cancer is that, one cancer cell and the other cancer cell next to it may not be exactly the same. We know cancer is heterogeneous and that there may be subclones within any one nodule or any one sample of cancer. So not even all of the cells may have the same alterations that you're looking for. So that's something else to keep in mind. And actually, very few platforms are really good enough to be down to 1 or 2% reproducibly. So what your limit of detection is plays a role into what is acceptable tissue. Now here, not only could you do something where you keep the architecture intact and can identify the tumor cells, but almost any platform would have enough, you have enough representation of the neoplastic DNA. I mean, even Sanger sequencing would be acceptable for this type of tissue. Okay, so in terms of test development, again, it's how you look at the overall picture. You have to go through the test development process just like you would for any other test. So in that sense, it really is no different. Scale is always the big question when it comes to NGS. How do you scale up test development to deal with the sheer volume of data coming from your sequencers? How are you interpreting it? What is the pipeline? How is bioinformatics playing a role? Building that bioinformatics pipeline is key, and something that becomes an even larger, significant portion of test development. So again, it's that scalability that comes up. But in a sense, in test development, you're still answering the same questions you would with any other method. What is your region? What method, what platform are you going to choose and use? Do you need to do whole exome or whole genome to answer your question? Is it something that you're going to select a small target region? And we didn't really even talk about the different capture or targeted methods for next generation sequencing. We just talked about the actual chemistry of the platforms without really going into the different techniques that are available for just applying this technology to a small part of the genome. So then as you move through development, you've answered some of those questions and you know what your method is, you've done your design, going back and testing samples and making sure that you can identify the mutations and the alterations that you want to detect, determining your sensitivity, your specificity, and so forth. Bringing in other questions, again, because of the scalability and the scale of NGS, looking at your workflows within your laboratories is something you start thinking about even in development. There's a lot of pipetting and manual pipetting involved. Can any of this be automated on liquid handlers? Becomes important and something that you want to start thinking about pretty early. Increasingly, you want to start thinking about clinical utility and reimbursement. This is not meant, clinical testing isn't meant as research. This is meant as trying to answer a specific question for a patient. And so what's the utility in this set of tests, in the tests that you're developing, in either the set of genes that you're looking at or the regions within the genes, and how much proof does it take to establish that clinical utility? Oftentimes, for particular drugs, these are tied with clinical trials, and how clinical trials are being designed and run is changing because of the availability and the impact of molecular data. So that comparison to the gold standard, your clinical trials, even that's changing. It's also always something to keep in mind. Data complexity, again, the importance in thinking of these things early, determining your workflow, determining the bioinformatics pipelines, the interpretation, and not to be forgotten, that last step, the reporting, finding a good, clear way to communicate the results and the significance of the results to the patients and clinicians who are going to use the data for patient care. Confirmation of results. For a long time, many places used FISH to confirm array data on the in the cytogenetic sphere. That's altered and fluctuated and decreased over time. Right now, that's to some extent where next-generation sequencing also fits in, perhaps maybe 5 years behind where array is or was. Again, regulatory requirements, mentioned CAP and CLIA and New York State before. Again, because this is clinical testing, this is all subject to regulatory standards of any other test. Because of the complexity and the scale, it becomes a bit more challenging perhaps to manage the scale of what's required. For example, we had a submission to a regulatory agency that was close to 1,000 pages for one test. There's also questions and challenges in how we handle intellectual property. as it becomes easier and easier to look at more and more genes at a time, or the entire genome or exome at once, how does that affect intellectual property that may be based or rooted in one particular alteration or one particular gene? And again, financial considerations. A couple years ago, the CPT codes for molecular and cytogenetic testing were completely revamped. And along with that revamp came a reevaluation of reimbursement, gap analysis, a whole process for getting new codes, and kind of changing that whole stacking code platform that we'd used for a long time. And so that really changes how reimbursement is looked at, not only by private insurance, but by the government agencies as well. So those are all things to consider. And again, because of the scale in large part, you can see being involved in next generation testing on the clinical side, and this is just our somatic team or individuals involved with different aspects of somatic testing here at Mayo, from the molecular genetics laboratory to molecular anatomic pathology laboratory, and then different members of core laboratories, bioinformatics, the Center for Individualized Medicine, et cetera. So it takes a team to put a lot of this together and to have it work on the clinical side. All right, so I know our target was to have a few minutes free, so for discussions and questions, it may not be 10 to 15, may only be 7 minutes free, but if there are questions or comments, I can certainly field some of those, but that's the bulk of what I had put together for everyone.
Thank you, Dr. Miller, for a very informative presentation. We do have a few minutes here for questions, and a reminder to all of our participants, you can submit written questions by typing them in the webinar's control panel. And at this point, I do see at least one question here. Question was from Irene, and it is, as more NGS tests are used in the clinic, how will the role of the technologist change in the clinical lab? Do you predict job loss for technologists?
I suspect there will be a changing of function. It's something that we've looked at in our laboratory as well, and we have technologists performing different functions within the laboratory. And so let me actually scroll back to that one picture. It's a really good question, and I think this is something where, and it comes up with any disruptive technology, there's a big question. How do you manage that change? And I think this graphic really kind of answers, or at least starts to address that question. The amount of work doesn't change. What the components of that work rearrange. And right now, by far, the bulk of the technologist's time is dedicated to the generating of the sequence, or maybe a few years ago was dedicated to generating the sequence itself on, by Sanger sequencing and reading it manually and all of that, reviewing it in various, in the chromatograms. Well, that's changing. As you can see, that purple piece is getting smaller. And so I don't see that as a problem. I see it as, well, look at the other pieces that are growing. At what point do our technologists become, individuals who participate more in either the data management or the data reduction or the bioinformatics pipelines? Because you visually can't review all the data anymore. It's generating way too much data way too fast. So you rely on some of the bioinformatics pipelines to do some of that for you. But you still need a human being to put a lot of that together, whether it's doing the actual work through the bioinformatics pipelines or starting to perform some of the downstream analysis, managing other parts of the process. So what I would see is some of the changes would be involved in how we start using technologists to participate in some of these other colors, either in the design, in the collection, or in the downstream analysis.
All right, do we have any additional questions for Dr. Romella? Remember, you can type them into the questions portion, or the chat portion on the sidebar on the right-hand side.
Looks like there are two more in there, Jason.
I'm scrolling. Here we go. Okay. Here is one. We have a question on, can Can we get trained in bioinformatics? How much computer sciences background do you need for it?
That's a great question, and that depends where you are and what level of bioinformatics you want to be involved in. There are obviously undergrad degrees and different programs that one can participate in. I don't know that that's going to be required for everyone. A lot of it will depend on the institution that you're in and what kind of, or what part of the bioinformatics you're participating in. Right now, there's also a lot of resources and pipelines that are already built, so it's more a matter of running them or interpreting them. So starting to put together data from clinical sources, interpreting literature, and that's all something that's kind of in flux. We don't really know for sure what level of training you need to have prior to working on these versus being able to run them or build them. Obviously, if you're building some of these pipelines, you need to have more programming and more of that background. But once you're running the tests or looking at some of the interpretation steps, it's not clear to me what would be required in terms of degrees.
All right, we got one more question from Troy. The question is, has the informatics pipeline improved significantly or sufficiently to handle the exponential growth of NGS testing?
It's a fantastic question. I would say the pipelines are exponentially improving. I say that very tongue-in-cheek because there are still improvements to be made. Nothing is, I think, locked down, and we're learning more and more as we go. So on the research side, the pipelines are continually improving. The challenge is getting them to the point on the clinical side where you can lock down, verify, and validate a test for a clinical practice. And so that's where you need to find a level in the pipeline that you can, you have the confidence and you can determine the sensitivity and specificity and the precision and the reproducibility and all these metrics that you need to be able to produce in order to generate a clinical test. And for some of these questions, I think that does exist. It's something where the pipelines have evolved to the point that they can be used in this way. Do I think there's still improvements to be made? Absolutely. I think there's a lot to do. And a lot of the bioinformatics pipelines that we have right now are primarily focused on the data reduction and data management, kind of the reading of the reads. But that orange bar at the bottom, that downstream analysis, looking at the differential expression, the regulation of the pathways within the cell, not the regulatory QC field, but the regulation within the cells, cell signaling, pathways, all of that, I think we're really just touching on it. Actually, the literature is just starting to explain some of these things. So we're going to be trying to synthesize existing literature and new literature for a very long time. So I think those pipelines definitely have a way to go. That part is still, that part of the orange or the light yellow on that graphic is still somewhat small.
All right, if we got time for one quick question, there's one more here. It's from Faith, I think, and it says, how close are we to detect incidental balanced rearrangements by NGS?
Interesting question. Okay, so it depends, again, on the platform that you're talking about, because if you have a DNA sequence that has a rearrangement, you can sequence right across that breakpoint and read that, which is a little different from some of the array platforms where it's still really difficult to see a truly balanced rearrangement. So you start getting back to how you're identifying the DNA or the RNA that you're sequencing. If you're doing a poly-A tail that's capture base, where you're taking down the poly-A to look at the mRNA, it would depend in large part on the quality of that RNA and how far from that poly-A tail that breakpoint is. Or if it's even being expressed in the cell type that you're looking at, that your sample is from. Something that could be rearranged but not expressed in any one particular cell, you might not see by RNA or by messenger RNA. If you're looking at DNA from any cell, it would depend on how you're capturing it. And the technology. Is it mate pair sequencing, where you're looking at specific pieces that are being put together? Are you looking at whole genome? Are you capturing particular genes so then you can see what's attached to those sequences? So depending on your platform and your strategy, you could identify balanced translocations, yes. But not all strategies and not all platforms will be as good for that indication.
All right, thank you. We are at the end of our time here. I have a couple more announcements to make here, but thank you, Dr. Ramilla, for your content presented today, and thank you for all of our individuals participating in this webinar. Within the next few days,
for your attention.
Within the next few days, webinar participants will receive an email from AGT that includes a link to an evaluation. Please take a few moments to assess this survey and provide your feedback on today's session. The webinar today was recorded and will be posted on the AGT website in the near future. We hope that you will join us in upcoming webinars over the next few months. We will send out a notification to members once registration is available for our next webinar. We hope to see you all at AGT's 41st annual meeting in Orange County, California, scheduled for June 23rd through 25th, 2016. Registration will be open in early 2016. Thank you again all for attending.